Hi, Dr. Javed Akhtar Ansari. Welcome everyone in this video lecture. I am going to talk about gene mapping. After watching this video, you will be able to understand what is gene mapping, what are the different types of gene mapping, genetic mapping, and physical mapping. What is gene mapping? Gene mapping refers to the process of determining the location of gene on the chromosome. So, gene mapping or genome mapping describes the methods used to identify the location of a gene on a chromosome and the distance between genes. The eukaryotic chromosomal genetic map is linear, whereas the genetic map of prokaryote is circular. So, what are the different types of gene mapping? There are basically two types of genome mapping to construct the maps. One is genetic map, also known as linkage map. Another one is physical map. So, what is genetic map? It tells about the relative or approximate distance or the position between two genes based on the genetic linkage information, but exact location cannot be determined here. In physical mapping, it tells about the actual distance between two genes on chromosome in terms of the numbers of base pairs between them, and this is the common method. Physical mapping further divided into three major parts, that is restriction mapping, FISH, STS mapping, and restriction mapping again divided into fingerprinting method and optical mapping. So now what is the genetic mapping and linkage mapping? Genetic maps and linkage maps, that is approximate distance of genes. So genetic map looks at how genetic information is shuffled or exchanged between chromosomes or between different regions in the same chromosome during the meiosis, which is a type of cell division, and this is the process called recombination or the crossing over. See here, genes are being shuffling here. Here the green type of colors and here red colors are there. Now these chromosomes are being shuffled in child 1. See here, the different bands are, it is expressed in different bands here. So here also, if you see, the different colors are there, and if you see this figure here, A, B, C is there. Here also a small A, B and C pairs of chromosomes are there. So chromosome crossing over taking place here, and that is called as recombination. So what happens here, if you see the colors have been changed over here, A, capital B and small c here, while here capital A, capital B and capital C is there. So capital A, capital B and capital C is replaced with the small c. Here again the small a, small b and the capital C. So this is known as recombinant chromosomes. Another concept is, see here the two genes are located far away and here two genes are located closer. So when genes are located far away, the chances of crossing over is more, and when the genes are attached or they are situated closely, so the chances for shuffling is less. So by finding out how often various characteristics are inherited together, even after the crossing over, it is possible to estimate the distance between the genes. So gene linkage, when more than two genes are situated closely on DNA called linkage group. See here the DNA, and genes are A, B, C, D, different types of genes are located closely. So this is known as gene linkage. So these are the two different chromosomes, capital A and capital AB and small AB. So this is known as sister chromatids. So after crossing over, one meiosis with a single crossover, what will happen? So there will be crossing over taking place. If you see here, colors have been changed. This capital A, capital B, capital A and small b. Whereas here the capital A and capital B was there, but here it is been replaced with the red color and small b. Here also, if you see, the A is as it is, but this portion has been shuffled with the capital B. So this is known as the recombination or shuffling. So suppose, for example, Here the shuffling taking place, the final product will be, suppose for example 40 recombination have been formed over here, and 40 non-recombination taking place over here, and 10 recombinants formed here, and here also 10 recombinant formed, and again here 40 non-recombination taking place. So because we are estimating out of 100 individual, for example we are taking, though here it is mentioned for the one chromosome, single chromosome, but here we are taking, to understand, for better understanding, we are taking the example of 100 individual. So this 40 and this 40 will be known as parental. The non-recombinant, they will be known as parental, whereas this 10 recombinants and these 10 recombinants means 10 plus 10 is equal to 20. So these 20 will be known as recombinants. And the formula for recombinant frequency is recombinant type divided by sum of all combinations into 100. So here if you see, the recombinant type is 10 plus 10, 10 plus 10 is 20, and the sum of all combinations 40 plus 10 plus 10 plus 40. So 40 plus 10 plus 10 plus 40 into 100, that will come 20%. So that is 20% recombinants and 80% parental. And the recombinant frequency is proportional to distance between gene. So gene map will be 20 units. So distance between A and B, distance between A and B will be 20 units. So that is 20 units, that is the chances for crossover will be 20 times. Thus, by following several traits through generation and recording how often the recombinants occur, one can map the relative position of corresponding genes. Then comes physical maps, that is the actual distance between two genes. Physical mapping gives an estimation of the physical distance between specific known DNA sequence on a chromosome. The distance between these known DNA sequence on chromosome is expressed as the number of base pair between them. So there are several different techniques used for physical mapping. These includes restriction mapping by fingerprinting method and optical mapping method. Another one is fluorescent in situ hybridization, that is FISH mapping, and sequence tagged site, that is STS mapping. The restriction mapping is a method used to map an unknown segment of DNA by breaking it into pieces and then identifying the location of the breakpoints. So this method relies upon the use of protein called restriction enzyme or restriction endonucleases, which can cut or digest the DNA molecules at short, specific sequences called restriction sites. A restriction map shows all the locations of that particular restriction site, that is GAATTC, throughout the genome. Restriction enzymes always cut DNA at a specific sequence of DNA. For example, the restriction enzyme E. coli R1, Escherichia coli R1, always cuts at the sequence GAATTC or CTTAAG, that is palindromic sequence, in which the nucleotides are arranged symmetrically in reverse orders. See here GAATTC and here GAATTC. So this E. coli will cut over here. Therefore, if we use E. coli R1 to cut the DNA, we know that the DNA sequence either side of the cut will be at AATT. And the steps involved in the restriction mapping: the collection of DNA sample, then the apply of restriction digestion single, then restriction digestion double, and then followed by agarose gel electrophoresis, followed by DNA pattern or band visualization, band analysis, and restriction map generation will take place. With this, I conclude my lecture. Hope you have understood the topic. We'll meet again with more interesting video lecture. So keep watching the videos. Thank you.